Hi, I'm DeLeo. I'm a faculty of biology at the Hopkins Station of Stanford University and senior fellow of the Woods Institute for the Environment. I'm thrilled to be here today for the ICI3D DAIDD 2021, the clinic on dynamical approaches to infectious disease data, to share with you some of the work of my team on integrating data and models for schistosomiasis control in Senegal. Before I move forward with that, I want to acknowledge upfront the wide network of amazing collaborators, of agencies, of donors, institutions that provided a crucial contribution to this venture. I cannot say enough to thank them all, and I can definitely not list them all, it would take too long, but I want to acknowledge this is truly a teamwork. Now, let me show you how is the structure of this three-part presentation. In the first part, I'm going to introduce you to the social and environmental determinants of parasitic diseases of poverty, and I want to tell you why we're working in Senegal, why Senegal is such a compelling place, at least for studying schistosomiasis. Then I'm going to talk to you about precision mapping of risk of transmission for schistosomiasis. And third, I'm going to talk about biological control of schistosomiasis transmissions and how we can integrate, again, data and models to make projections of what might work, might not work for the control of schistosomiasis. Let me start then with the first point. Well, thanks to the rampant vaccinations, and we start to see some light at the end of this devastating and tragic COVID-19 pandemic that just today has exceeded 271 million certified cases worldwide and is approaching to 5,300,035 deaths. There is a silent epidemic, though, that has been going on day after day, actually for centuries, with no reliefs, affecting nearly 40% of the world population. Today, no less than 3 million people typically living in tropical and subtropical countries with limited access to clean water, to healthcare and nutrition. And I'm talking here about, as you can see, a family of diseases caused by pathogen and parasitic worms that share three features. The first one, they all have an environmental reservoir, whether it is a free living stage of a parasite or an animal reservoir. For this disease, contagion occurs from infected bites from vector, for instance, such as mosquitoes or ticks, or simply by stepping into contaminated soil or water. These are also diseases of poverty, affecting disproportionately population with limited access to resources and healthcare. In general, for this disease, there's no long-lasting immunity in a lot of cases, and there are certainly no vaccines, or the vaccines that are there are too expensive or provide only partial immunity and for only some a temporary time. So sick people can be treated, and in a lot of cases can be treated quite effectively, but treatment does not protect from the infection. Therefore, targeting also the environmental reservoir and preventing exposures are two key approaches to fight this disease. And the goal of my talk is to show how digital technologies can help us to address this problem. I want to show you this by using one of the most important of the so-called neglected tropical diseases as a reference example. This disease is schistosomiasis. It's a debilitating parasitic disease of poverty. It's endemic in 4 continents and 74 countries in tropical and subtropical region, and affect more than 200 million people, the vast majority in sub-Saharan Africa. It is caused by a parasite of the blood vessel. This parasite has a complex life cycle. It requires 2 hosts, the definitive host or human host, and a limited set of freshwater snails strongly associated with different species of the schistosoma parasite. And these snails works as the intermediate host of the parasite. These snails are usually quite tiny, as you can see here in this picture. And they look harmless, but they are hijacked by the parasites, parasitic castrator, in fact. They transform them in a factory of infectious stages that are called cercaria, that are released in the water for several weeks after infections. And what actually happens is that people get contaminated by stepping into water where snails and cercaria are present. We can treat people And there is a drug, in fact, that is effective. That drug is called praziquantel. It works, it's cheap, but after removing the parasites from people, it does not do anything to target the environmental reservoir of the disease. So snails continue to shed infectious stages for weeks after drug treatment. And like on a treadmill, this leads to an endless cycles of treatment and reinfection. To interrupt these cycles, the WHO has recognized that schistosomiasis can be controlled only by completing medical treatment with environmental intervention. And because we always operate in conditions of limited resources, it is actually crucial to identify and target snails and snail habitat, to have a more effective control of schistosomiasis. To show you how we can do that, I want to bring you in a quick journey to our field sites in Western Africa. So we are here in Senegal. We are in the lower basin of the Senegal River at the border of Mauritania, where we've been working now for almost a decade. And the reason why we are here, which brings me to my second point of the introduction, is that in 1986, the joint ventures of 4 countries in the area, Senegal, Mauritania, Mali, and Guinea, get together and complete the construction of the Jama Dam, which is a dam to prevent saltwater intrusions in a fertile lower basin of the Senegal River. And the dam has been actually very effective in doing so. But unfortunately, a year after, it started to report cases of urinary schistosomiasis, which was endemic in this area before the construction of the dam, but also of intestinal schistosomiasis, which was not even present before the construction of the dam. And this has led to one of the most spectacular outbreaks of schistosomiasis, which is a little bit of a paradox, because in contrast to diseases like influenza or even COVID, of course, schistosomiasis is a parasitic and chronic infection, and usually does not lead to this boom and sometimes maybe best of these oscillations and this rapid increase of transmission in this area. In some of the villages where we work, like this village Malatak on Lac de Guiers in Senegal, there is a 99% prevalence of schistosomiasis, 90% of Mancini, 98 of hematobium, the 2 major species that cause schistosome infections, and with a lot of co-infection. The reason why that happened becomes kind of clear as soon as you look at this footage that is done by using Landsat imagery in the last 30 years. And you can see that we're going to recover from 1986, just before the construction of the dam. In the year later, you see how the landscape has changed. And what actually happened is that more water has led to create better habitat, freshwater habitat for the snails, and led more snails, an abundance of snail population, and then more disease. We've been investigating the complex network of cascading effect in some papers that are related also with the development of agriculture activities that are associated as well also with an increased risk of schistosomiasis. Now, the question here is that whether we can maximize positive health outcome in the face of limited resources for surveillance and control. And the point is that in this heterogeneous landscape that we see there, whether we can identify the hotspot of transmission for schistosomiasis, where we can deliver best praziquantel and possibly also environmental interventions. So this part of Senegal that has become the most hyper-endemic areas for schistosomiasis in the world. And we're lucky enough to lead 7 field missions over 16 study villages, basically tracking levels of schistosomiasis infection in about 1,500 school-age children and treating them and looking at different type of intervention there. Our work, in addition to the human parasitology, and also significant malacology component. To this end, we have spent countless hours collecting snails from the river and lakeside places where people congregate for fishing, for bathing, and other daily tasks. We have collected nearly 15,000 snails and meticulously recorded data about all of them, from the exact spatial location they came from, along with the type of vegetation they were found, to precise measurement of the shells, the shell size, and of course, which parasites lived inside them. This is what we found. We found a very patchy distribution of snails, surprisingly for anybody who work on snail-transmitted diseases. We found a strong association of the snails with certain type of aquatic vegetation, typically Ceratophyllum and Potamogetum in these areas, as we documented in a paper analyzing the distribution of snails. But in some cases, we really had a concerning mismatch and unexpected. There are cases in which we found non-ear short snails in spite of high infection levels in children. So there was something out there, and we realized that we really needed to change perspective, literally, in this sense. And so back in January 2017, we got a simple recreational drone with an RGB camera, and we started to take into images and map our field site. And what we can look with this bird eye view at about 50 meters height in our field site is that, wow, there was a lot of floating vegetation that we knew was associated with the snails that amplified schistosomiasis. And it might be that in some cases there are not a lot of vegetation near shore because people tramp into the water, remove it. They don't want to have the vegetation, they want circulation, but they can't really do a lot for where there is deeper water. And not only we could see there was a lot of vegetation, but just visually we could start to map the vegetation that we knew really well. So we could see the emergent vegetation like the Stifa and Phragmites, identify area of open water where we didn't find snails in general, and also areas where we had Ceratophyllum and Potamogetum in huge abundance. So in conclusion to my first part here, the point is that led us to a research question, actually, whether precision mapping on suitable snail habitat could have helped us to identify transmission hotspot. And so I'll stop here, and we're now moving to the second part of my presentation.